Have you ever found yourself wondering what an examiner is really thinking when they're marking your paper? Well, today, we're going to pull back that curtain. We're taking one single exam question and breaking it down piece by piece to show you exactly how to score those precious marks.
I mean, it's the ultimate question, right? You've spent months revising, you feel like you know your stuff, but then how do you get that knowledge onto the page in a way that just screams, give me the points? How do you give that examiner exactly what they're looking for?
That's what this is all about. We're going to decode a real question from an AQA higher tier biology paper. And our secret weapon? The official mark scheme. This isn't guesswork. These are the examiner's own rules for the game. All right, so here's the plan. First, we're cracking the exam code. Then we'll hit the photosynthesis fundamentals. After that, we'll get into the why of the experiment, analyze some data, and then wrap it all up with the key takeaways. Sound good? Let's dive in. So our subject today is question 6 from the June 2023 higher tier paper. It's all about photosynthesis, and it really covers the whole spectrum, from the basic equation all the way to experimental methods and data analysis. Okay, let's kick things off with the basics. This is a 1-mark question designed to test your core knowledge. It's all about recalling one of the most fundamental equations in biology. The paper gives you the bare bones of the symbol equation for photosynthesis. You can see the formula for glucose is already in there. Your job is simple. Fill in those 3 blanks with the right chemical symbols. And here we go, straight from the horse's mouth, the mark scheme. You need H2O and CO2 on the left, the reactants, and O2, the product, on the right. But check out that little note. This is an insider tip. It says the order of the reactants doesn't matter. So CO2 plus H2O is just as correct. It's a small detail, but it shows you there's often a bit of flexibility. So that's the basic recall done. But you know exams are about more than just what you know. They're about applying that knowledge. These next questions are all about understanding why scientists do things in a certain way. It's all about the experimental method. So here's the setup for the experiment. You've got a lamp for light, a beaker with these little leaf discs, and they're floating, or sinking rather, in a sodium hydrogen carbonate solution. The whole point is to measure how long it takes for them to make enough oxygen to float back up to the surface. Let's see how the questions pick this apart. First question, why bother getting all the air out of the leaf discs before you even start? This is all about controlling your variables and making sure your results are actually valid. What's the real reason here? And look at this. The mark scheme actually gives you a few different ways to get this right. You could go for the simple practical answer. You do it so the discs will sink. Or you could get more scientific and say it's to make sure the only gas making them rise is the oxygen from photosynthesis. Any of these would get you the mark, which is a great thing to remember. There's often more than one correct way to phrase your answer. Okay, next up, the solution itself. Why not just use regular old water? This question is a direct test of whether you know the ingredients for photosynthesis. What does that plant need that plain water just doesn't have enough of? A classic 2-mark explain question. For these, you always need 2 linked ideas. Here's a great exam technique. Think what and why. So for mark number 1, you state what the solution provides. That's carbon dioxide. For mark number 2, you explain why that's important. It's needed for photosynthesis. Boom. A perfect 2-mark answer. So what's actually going on here? How do these little leaf discs magically float to the top? This question is about connecting the product of photosynthesis with what you actually see happening in the beaker. And again, it's a 2-marker, so we need 2 connected points. First, you have to state the obvious but crucial fact: oxygen was produced during photosynthesis. Second, you explain that this gas gets trapped inside the leaf disc, which makes it less dense and causes it to float. See how one point leads right into the next? That's the key. All right, let's move on to the data analysis. This is where things get a little more complex. These questions test your ability to not just read information, but to really interpret it and apply it. First things first, we gotta identify the variables. The independent variable is the thing you change on purpose. In this experiment, that's the color of the light. The dependent variable is the thing you measure to see the effect, and here, that's the time it took for the discs to rise. Nailing these down is absolutely fundamental to understanding any experiment. Now for a question that makes you do a bit of biological thinking. We know plants have different types of chlorophyll, like chlorophyll A and chlorophyll B. So what's the point? Why is having more than one type of this pigment a good thing for the plant? The answer is so the plant can absorb a wider range of light wavelengths. But wait, look at that extra note.
This is a golden nugget of exam wisdom right here. The mark scheme explicitly says do not accept to absorb all colors. This is such a great lesson in scientific precision. More or many is accurate. All is an exaggeration, and so it's marked as wrong. Examiners are always looking for that kind of precision.
And here we are, the grand finale. The last question is worth a hefty 4 marks, and it asks you to connect 2 different bits of information: a graph that shows which wavelengths of light chlorophyll absorbs, and a table that shows how fast the leaf disks actually rose under different colored lights. You've got to be a detective here. So, how in the world do you bag all 4 of those marks? Well, the mark scheme lays out a perfect roadmap. Point 1, state that blue light is absorbed really well. Point 2, make the opposite point, that green light is absorbed very poorly. Point 3, link that absorption directly to the rate of photosynthesis and the time it took the disks to rise. And now for the final killer point. You must quote data from the graph to back up your claim. Something like, about 80% of blue light is absorbed. It's all about making your point and then proving it with the evidence they gave you.
Whew. Okay, so after taking that entire question apart, what are the big lessons? What can we learn from looking right into the mind of the examiner through the SMART scheme?
Here are the 4 biggest takeaways. Number 1, precision is king. Be specific, use the right terms, and don't exaggerate. Number 2, understand the why behind every step in a method. Number 3, for those big mark questions, you have to connect the different bits of data. And finally, number 4, remember there's often more than one path to the right answer.
And that just about wraps it up. I'll leave you with this question to mull over. Hopefully, taking this deep dive has demystified the marking process a bit and given you some new tools to think like an examiner. Thanks for tuning in, and I hope you found this useful.